Thank you very much. Thanks for inviting me here again, actually. I came to the Berlin meeting, which was great, a couple of years ago. And I'm a bit embarrassed that I can't be here for the whole meeting this time. The schedule just looked amazing to me, because I'm really here to learn on these conferences. I work on, as you're about to hear, on colour and vision in nature. But of course, we don't always know how the brain works and how the visual system works. And I think some of the things you're doing here help me to sort of fill in some of those gaps. So it's been really amazing, actually, to hear speak with the people I've spoken with so far. And hopefully you can help me out after hearing this. So to set the scene, we go back to the origin of life on Earth. And the Earth formed about 4.6 billion years ago. The first life forms came around about 3.9 billion years ago, but they were simple single-celled bacteria-like things with no nuclei. The first cells with a nucleus appeared about 1 billion years ago, and they began to clump together. First of all, they didn't interact. They formed balls of cells that just moved around in the same direction. But then they began to interact and tissues started to develop. By about 550 million years ago, we had all the phyla that we have today on Earth. So all the major groups of multicellular animals that are classed as phyla, they all appeared by about 550 million years ago. But they didn't look anything like what they do today. They look more like this. This is a scene from the Ediacaran fauna, fossils that we found particularly well represented in the Ediacaran Hills in South Australia. And you'll see that these animal forms, which they are soft-bodied, they move around slowly, and they don't interact with each other in any way. Life is fairly slow. But all that time, it was also black and white. It wasn't really black and white, but there was no adapted colour. However, all that time, sunlight existed. So you had sunlight falling on all these animals, but nothing had reacted to it. Possibly because a reaction would take up so much energy, and nothing could afford that. They ate in such simple ways, they didn't interact with each other, so that perhaps they just didn't need to use the sunlight. In fact, it was used to some extent, for example, to know which is up and down in the water column, but not in terms of vision. They didn't see images of each other and interact with each other through that means. An interesting thing about sunlight, of course, we all know that this is the sun spectrum. We've got there the peak in this here. This is under different cloud conditions, so that's no cloud. That's with high cloud, and that's with no cloud. And you see the peak, as you expect, between something 350 nanometers to 700 or so nanometers. And then, interestingly, you've got the transparency window in the sea mirrors that. You've got a region of of transparency here that corresponds to the peak in the sun spectrum. So that's really nice. You've got this this range of the human visible range, if you like, that's just perfect for vision. And indeed, vision happened. This took place about 521 million years ago in this animal here, a group of trilobites. So this is sort of that sized animal. It was a predator, and it was a fast-moving animal. So suddenly, it's got this pair of great eyes, compound eyes. You can just see one of them sticking up here, casting a shadow. They're quite large things. They saw really, really well. And we have really good fine details of some related species to these, where we can see all the innovations inside the tiny lenses. So they form this perfect vision. Now, to let this animal loose at that period of time, when everything else is soft-bodied, everything would have suddenly appeared as chunks of protein just waiting to be eaten by this animal. So life responded. There was a great evolution took place. I like to see, consider this actually, because this is the difference between biologists and my physics colleagues here on the bottom. What we had all these wavelengths at one time in the water. Life was exclusively marine at this point. But then with the first eye, you've got this picture. So you go from this bunch of wavelengths to suddenly this picture. Suddenly you see this, the animals were using this entire spectrum and seeing in colour, we believe. So what happened? Well, there was actually an explosion in evolution, something that actually is called the Cambrian explosion, or the Big Bang of animal evolution, where all these soft-bodied groups evolved hard parts and complex lifestyles. They became predators and prey. They had hard parts for swimming, so they could escape these new, highly mobile predators. And they involved eyes themselves. Here, for example, they started to use colour. Where does colour begin? Well, in terms of pigments, for example, here is a polychaete worm today that is totally maladapted to colour. This is a chunk of protein that's advertising itself as bright orange, but it would never be seen in the environment like this. Actually, this animal lives in an opaque tube. So if you pull the animal out of its tube, it looks like that, and that's the starting point for evolution. It contains chemicals that are part of its physiology that would go on to be adapted through natural selection. For example, in this case, you might get these orange pigments go on to evolve to become brownish for camouflage or greenish. So here we are at the end of the Cambrian explosion. This is a typical scene in the Cambrian period, about, in this case, 508 million years ago. And you can see now there's these different groups of animals that are starting to now look more like the animals that we see today. There are groups from the arthropods there, from the polychaete worms, and some other groups. And they were starting to use colour. And in fact, they were using colour for just the same things that animals are using colour today. And as I said, they also evolved eyes. Here's just one example of a Cambrian animal over 500 million years old with very, very good vision indeed. Here's this animal. It's quite small, but this is its head, all this region here, and that is its eye. So it's got an eye that's about a third of the size of the whole body, but it's preserved really, really well in fine detail. These are scanning electron micrographs showing the cornea region, and below the smooth outer surface, we've got this fine network of fibrils that allows for transparency. And then in the center, this is a cross-section in transmission electron micrograph, and we've got this layer that would have been continuous, and this is a yellow pigment, so that the animal could have distinguished between bioluminescent prey and sunlight. We've got evidence of animals using bioluminescence, making their own light in the Cambrian. As soon as this first eye evolved, everything began to happen, everything we find today in terms of visual adaptation. So this is a yellow filter in the eye. As I'll show a few examples later, but some animals use bioluminescence to camouflage in water, because in some parts of the water, sunlight is exclusively blue, and blue travels best through sunlight. So bioluminescence in the sea tends to be blue as well. But some animals use it to actually shine light downwards to prevent acting as a silhouette from any animals passing below. And the yellow filter will actually pick out the slight differences in wavelength between sunlight and this bioluminescent light. So there's bioluminescence at the top. I mentioned, I separated the ways that animals produce colour into 7 different mechanisms, or colour factories if you like, because you've got sunlight falling on it, or sometimes making their own light, but you've got lots of little moving parts involved, tiny little factories there that are affecting the light rays in various ways. I'm going to really concentrate. Last time I spoke about all the different ways of producing colour, the pigments and so on. But I'm going to today concentrate, for a reason, on structural colour, which is the thing I've been working on most recently. So this is the purely transparent materials that interact with light rays to produce some of the brightest colours that we find in nature. And the origin of this subject goes back to perhaps Isaac Newton or Hooke, when in 1704, in his book Optics, he showed that indeed the peacock feather must contain a series of thin films to produce these colours. For me, actually, I started it in 1990, looking at this animal from Australia, which was very interesting at the time because it was something called a photonic crystal fibre. And the idea of a photonic crystal was only just invented in physics in 1987. And there were Nobel Prize winners, and it was just this amazing thing, this 2D or 3D structure that can manipulate light at the single wave scale. And when I was looking at these animals, they were filled with them. I started looking around at all sorts of other marine groups, and they were filled with them as well. So nature had been evolving these amazing technological optical structures over, in fact, millions of years. And we'd got excited because we'd just thought about it a few years ago. I also in my early days, came across lots of cases of diffraction gratings, which are, in this case, quite simple. They're just surface structures that split white light up into its component colours, just as you would find in a spectrometer. But I found that they were having a behavioural function. These are the antennae of a ostracod crustacean, which is a tiny little animal. Typically, it's about the size of a tomato seed. It has a shrimp-like body that's totally enclosed within a bivalve shell. And these antennae are hidden within the shell, except during courtship, during mating. So I filmed these at the bottom here. These are frames from a video recording of two seed shrimps mating. We've got the female at the top and the male below. These animals only see within about 10 times their body length. But when the male does get within view of the female, it puts these iridescent hairs outside of its shell, and the female becomes receptive and they mate. So it's working as an efficient courtship response. But when I looked at the evolution of this group over 350 million years, we found that the optical structures were improving in efficiency through that whole sequence. Not only that, so too was the eye. In fact, at the beginning of this, we have the iridescence evolving and then an eye evolving, and the eye began to increase in efficiency too. And it's been a really successful mechanism because these animals have become the most abundant metazoans, multicelled animals, at least in Australian waters. But then the oldest gratings that we have go back to the Cambrian period. The famous Burgess shale fossils from the Rocky Mountains in Canada, 508 million years old, I looked at about 50 different species and found diffraction gratings in these three here. If we take a look at that one there in the top right, the fossil, part of the fossil looks like this. It's really well preserved. You've got this, it's a relative of polychaete worms. And here's its body with lots of spines coming out the side. But if we dissolve away some of the rock with acid, we get the original organic material still preserved. And we can find the grating structure preserved really well. And it looks like this under white light in seawater. So these were the actual colours that were shining 508 million years ago, soon after the very first eye evolved. And new scientists made some nice colour reconstructions. They got quite excited that this was the first colour reconstruction of an ancient extinct animal. But these are really, you know, they're true colours that would have been there in the sea. Perhaps to provide a warning colour of the spines that they had in this case. Now we did, in this subject of structural colour in nature, something that's been well known for some time is the idea of multilayer reflectors, thin films, stacks of thin films, as you can see in the the wings of the blue-winged wasps, or bees. So this is really well-known stuff. This is a single film, as shown here, has effectively two surfaces, interfaces between different refractive index materials from where light reflects. So just like a layer of oil on water, a fly's wing is a single thin film. So you get, when light comes in, you get about 10%, or let's say about 5% reflected from one surface, the rest travels through, another 5% reflected there, and the rest travels through. And these two reflected rays can interfere constructively or destructively, just in this manner. So where they interfere constructively, we see a bright colour for that particular wavelength. But you only get one wavelength working in that way for each angle of incidence. As the angle of incidence change, that condition works for a different wavelength, so we get a different colour. So you get a spectrum of colours in reflection as the angle of incidence changes. But they became quite intricate in nature. This is Sapphirina, a copepod crustacean that's only about 5 millimeters long. This is a male. The female is transparent. But the male, within its cells, has this nice stack of hexagonal platelets interlocking. These are made of guanine, a refractive index about 1.83, that are working essentially in cytoplasm, which is about 1.333. Now, when you've got a stack of layers rather than just one, that single one reflected about 5% from each of those layers, so you've got about 10% reflection. When you've got about 10 layers, you get 100% reflection, total reflection of all the light. So it's an excellent reflector. And here, the change in colour is down to slight variations in the angle. So you've got different wavelengths being reflected constructively from different parts. And multi-layer reflectors, they turned up in fossils. I mean, these things are physical structures, just like a bone is a physical structure. So if the preservation is fine enough, these tiny structures, layers just 80 nanometers thick, will preserve as well. This is the head of a beetle from Messel in Germany. This is a famous fossil site now, but in the days when I looked at these beetles, it was about to become a rubbish dump. And then UNESCO made it a World Heritage Site. So it got saved, a reprieve at the last minute. But they've just got amazing colours. This is the head, this is the thorax and the jaws there. All of these are bright structural colours. In fact, you can break open the rock and it just looks like a living beetle. It's just, the colours are so vibrant. Because if those fine structures remain and light strikes it, then the colour will remain as well. I set about testing some of these, so here's some electron micrographs. These are the thin layers going here, a little bit crumpled, a bit wrinkled there. And this is looking at them from above. You can see they're nice and flat otherwise. A few layers are peeled off there. But the predicted reflection spectra is exactly the same as what we see. So that proves that we're looking at the right thing. Now, nature sometimes tells us something that we don't know. And this is something that's led to some really important commercial colour. These are swimming crabs called ovolipes from Australia. And they have a really nice iridescence. But they have lots of relatives that don't particularly have bright iridescence as well. And I'll come to that next. But if we take a slice through their cuticle, this is what's causing the iridescence. So this is the very outer surface, where there's a slight angle here, the cuticle. And these layers, rather than being flat, are actually wavy. You've got a sort of a slight sinusoidal arrangement going on, which means that the reflectors are actually angled slightly. And what that does, for each wavelength, it means that the colour will be reflected over a broader range. Now, this is the evolution of that group, and this is why that's important. They started off in evolutionary history using sound to signal to each other. They have stridulatory structures. And all of this group here, these species, just use sound only. Then they began to evolve some iridescence. These little patches here represent the areas of iridescence. Well, when they moved into deeper water, they started to almost cover themselves in iridescence. And the reason for that is that the red light will be reflected back up into the water, but blue light along the horizontal. Well, in shallow water, that would be too dangerous. The red light reflected up would have attracted their fish predators. They'd have been glowing like a beacon to these predators. But in the deeper water, where only blue light exists, you've just got blue light traveling along the sea floor. And they even have these undulations in the layers to broaden it, so the signal is quite wide enough for to attract the attention of one of their own species. So that's really nice. It's great for the crab, but it's great for us too, because we've managed to copy that now and make new forms of structural colour that only appear as a single colour. So before I was talking about those rainbow effects and the spectrum that you get from within a spectrometer grating, for example, well, it's almost like taking one of those colours and stretching it out, almost removing the other colours, so that from any angle you're looking at it, it appears the same colour. Effectively, what we've done is exaggerate what was going on in the crab. And suddenly, there are so many applications for this. We're trying to make it in pigment, in flake form, so that it can go into paints, for example. Well, one of the best studied animals for structural colour, well, the best group, is the butterflies. Their wings are covered in scales about 80 microns long that overlap like tiles on a roof. Now, these are the perfect devices to evolve structural colour because they, by nature, contain components that are around the 100 nanometer scale, which are ideal for structural colours, for reflecting light. So here we've got a close-up of one individual scale, a typical butterfly scale, and these have evolved into a whole range of forms. These are just a few examples. So these are scanning electron micrographs of just part of a single scale in each of these butterflies. And by varying these different components in different ways, you can get different optical effects. And this tells us now, when we've got our basic structure that we've copied from our crab, we can adjust that slightly in the same ways as we're finding here to produce the same types of colours that's going on there. I mean, I can show you, I have one sample with me actually only, but one interesting thing is that is there going to be some kind of difference in the way we see this type of colour compared to looking at a pigment of the same hue? So something, whereas these structural colours are reflecting the same wavelength of light over all directions, just like a pigment is, but there are a few other properties buried in this as well, a few slightly different effects. And is this going to actually affect the way we see it? That's an interesting question. Also, we have, with some structural colours, multifunctional properties. So as well as being, having these bright colours, they're also scratch resistant or hydrodynamic, or they repel water. For example, some of these butterflies that are producing really nice colour, when their wings are horizontal, we put a drop of water on their wings and the water just moves along and drops off the edge. It gets ratcheted along because there are super hydrophobic structures in there too. Blue actually is a colour that's turning up a lot in nature, and we've only just realised it. We're just putting out the paper now actually, that the reason for this is that it's really easy to evolve. Red is one we're having problems with, but blue actually is all over the place in nature. And in fact, it's because it's not a narrowband colour after all. We first thought that blues were in nature were the result of Tyndall or Rayleigh scattering, where the longer wavelengths actually just miss the structures and the shorter wavelengths hit them and get reflected. So you get a lot more reflection of the shorter wavelength blues than the longer wavelengths red. But then we found that if you look at these structures, this is a cross-section through a blue bird feather. If you do a Fourier transform, there's actually a lot of order there. So they're acting like multilayer reflectors, as we saw earlier. But the question is, why then don't they reflect other colours too, and why only blue? Well, the reason is that they are reflecting a big range of colours. They're what's called broadband reflectors. So they're reflecting a large part of the spectrum, but with the blue, they can manage to center that into the ultraviolet and just take in a little bit of the blue at the end of that. So by having a broadband reflection, you can just catch the end of the blue region of the spectrum, blue and violet. And in fact, even green. Green is a colour that animals have real problem with. Animals don't seem to be able to have evolved a green pigment. And yet green is a colour that a lot of animals would want for camouflage reasons. So lots of reptiles and amphibians and lots of birds have evolved green through a combination of blue and yellow. Yellow is a pigment. That's a pigment they can evolve really easily from their food. And blue is the structural colour. So here's the green tree frog of Australia. It isn't even, it has the Latin for the word blue in its name, because when it was first discovered, it was put into a jar of rum, moved back to the UK, identified, and it was this nice blue colour. So they named it after the colour blue, ceruleus. But in fact, of course, this is just one part. The yellow pigment had leached out in the alcohol and left just the structural colour, which was perfectly preserved. So here's another case of colour mixing, for example. In this case, yellow, just the same principle. So if we shine these lights onto a board, that light is created through just using 580 nanometers, which we recognise as yellow. This light here is 540 nanometers, we recognise as green. This one here, 620 nanometers, as we recognise as red. So in this region here, there are no wavelengths for yellow light at all. Yet if we combine the two, we just see yellow. We get the opposite of colour as well, which is starting to prove interesting to us. We've got lots of cases of transparency. So to disguise yourself, particularly in the water, the larvae of lots of crustaceans have evolved complete colourless bodies. They have a little problem. They have a problem with the eyes. They can't camouflage those. Also, if they eat something, they're in trouble. But they've not only got transparent materials, but structures on the surface that prevent reflections too. Here's an example. This is an interesting one, actually, from... I discovered there was a population of Drosophila fruit flies in Japan that had been kept in dark for 56 years. And in fact, if we just look at these top two here on the left, A and C, this is the normal fruit fly cornea. It's got these tiny structures on the surface to act as an anti-reflector. It allows all the light to pass through its surface. So that's great for vision. It improves vision. You're able to grab more light, particularly if you're flying at dawn or dusk. But then after it had been in the dark for 56 years, the population's eyes look like this. They'd lost their anti-reflectors. But actually, if you then put the population back in the light for another year, they grew back again. In fact, strangely, they hadn't disappeared at all, but the spaces between them had been infilled. So here is a cross-section, and you've got these little nodules sticking up there. And this is a cross-section of one that's been in the dark, and the spaces has been infilled with material, probably to just add some extra strength to the cornea, now that these anti-reflective functions weren't needed anymore. So we get these what's called moth eye structures appearing on all sorts of things, particularly the transparent wings of butterflies. Another thing embedded in the transparent wings are polarization signals, things that actually we don't see, but other things do. And that's becoming important, actually, for in particular beetles. Now, at the Natural History Museum, we have about 10 million beetles specimens that we've got people working through them with spectrometers, looking at the actual colour signals. The colours produced by, it can either be a standard multilayer reflector, or it can be a more liquid crystal type arrangement of molecules, where the molecules are layered but twisted with each layer, just as in a liquid crystal. But what that does actually produces circular polarization of the light, something we're not sensitive to. But it's opening up a whole can of worms, actually, in this case, because these are a group of beetles here along the bottom. These spectrometer readings are from a single species of beetle, and they vary widely. And we're starting to find that to us, they all look identical. So things we've identified as individual species might actually now be broken up into lots more things, lots more groups, which would actually be a little bit of a problem for us because we've got too much work as it is. With about, I think we only know about 10% of what's already there. So we could be increasing that value quite a bit, thinking of the genetic variation that lies behind this. What these show here, these are the results for many individuals of each species. So these 3 species there, that's fine. They all produce the same mechanism. These different colors now relate to the different types of graphs. And you can see for many of the species, they contain a range of these reflection patterns. So what this tells us really is that there's a whole world going on around our very eyes we have no idea about. All these animals producing these private signals that we just don't know exist. We're just not picking up on them at all. And the way biology is at the moment, it doesn't factor this in any way. To make matters worse, we've also got ultraviolet to worry about, because most species also see in the ultraviolet region about 350 to 400 nanometers. Well, when I was looking at a group of crustacean parasites on marlin, this is the surface of a marlin, and this is one of those, what's known as a fish lice. They use chromatophores, which I'll talk about in a minute, that are filled with pigments to match the colour of the animal that they're on. Because fish can come along and pick off the parasites. There are cleaner fish that feed on these things. So they have to be camouflaged. But when I took an ultraviolet photograph, they absolutely disappeared. So they're also matched in the ultraviolet too. And you can continue this theme. There's ultraviolet patterns on flowers to attract bees. There are ultraviolet patterns on insects that they use to attract each other, but actually birds pick up on those as well. The amount of interactions going on in the ultraviolet world is just another signal that we don't understand, or we don't even comprehend what's going on around us. So we were a long way off fully understanding things in terms of vision. So those colours I've been talking about mainly are single wavelength colours. They're producing a single hue when we've got a stack of thin films like that. But if we actually make the layers different thicknesses, then each one of them will be responsible for reflecting a different wavelength in that same direction. And eventually all those wavelengths can overlap to form silver, a broadband colour, maybe even gold or copper. We're finding some of those now in chromatophores. It's proving interesting. Chromatophores it's almost like the holy grail of biomimetics in terms of colour. It's really to have a cell that changes shape and affects the colour, so you can change the colour of your material. We've been trying to copy it for years without great success, unfortunately. We're getting close. But a chromatophore, this is a single cell filled with pigment that can expand or contract. It's the same cell expanded and contracted. So when it's expanded, it's large enough to appear as a pixel. But when it's contracted, you can't see it anymore, it disappears. So you can imagine having red, blue, and green chromatophores packed together. By expanding and contracting them, you can produce any colour that you want. That's true to a degree, except that we're now finding that chromatophores are involving mainly yellows, oranges, and reds, and the rest is down to mirrors. This is one actually this is a blue, but actually it's not a blue chromatophore. This is the blue-ringed octopus, whose blue rings light up when the animal becomes aggressive. But the blue is a structural colour, and it's the background that changes. The background can be white normally, so the structural colour is reflecting the blue, but the other wavelengths are passing through, being back reflected. So the whole thing appears a bluish white. But then when the background, those chromatophores expand and the background becomes black, it absorbs all the rest of the colours, so only the blue is reflected, and it comes across as a really bright electric blue. And these are close-ups of chromatophores from many cuttlefish and squid. But again, you can see here it's starting to show mainly oranges, yellows, and reds. And just behind, we're getting these mirror effects to reflect the blues and greens. But these produce some amazing camouflage. They're just incredible. We can mix up our layers in different ways. This is a cross-section through fish skin, showing one of those broadband reflectors. It's silver, and indeed you can see in any cross-section there's a whole range of thicknesses of the layers. Fishes often use silver to be camouflaged in the water. So if the fish is in the water, you're looking at it, you see a reflection of the surface, so you believe that between you and the surface there's nothing in between. So effectively the fish disappears. But I think they can also be used to confuse predators. I've been within a shoal of silvery or blue metallic fishes, trying to catch them, and I literally can't. My eye is drawn continuously, subconsciously, from one direction to the other, and I find it impossible to follow a single individual. But when there's just one on its own, I can catch it straight away. So I think that there's a function going on here for using structural colour to confuse predators in fishes. And this is one where we've been studying the evolution. This is the blind Mexican cavefish, which began its life outside in open water with nice big eyes and a silvery body. Throughout evolutionary history, it's moved into a deep underground cave system. Today, we're finding populations further and further into the cave system. The further they are in, the longer they've been living in darkness. Well, instantly the eye has disappeared, but the silver's only gradually disappearing because it's not so energetically expensive. So by looking at how it's actually devolving, we can get ideas about how the colour evolved in the first place. And mirrors are used in bioluminescence as well. This is one of my seed shrimps again in a glass of seawater. You can see the individual here. And it's making these nice trails through the water. So this is in total darkness. There's no flash there at all. And these are hatchetfish that shine using mirrored tubes, the bioluminescence directly downwards. So you can see on the underside, you've got these little exit points for light. And we've got these series of tubes. So there's just one bioluminescent source, and it shines the light down these tubes, and they have these nice light splitters so that light comes out these exit points. And they get focused into a narrow beam as it passes through here. So the light only goes directly downwards. So when these animals are in mid-water, where only blue water exists, blue light exists, a predator below will see blue sunlight, and as it passes beneath this fish, it will see exactly the same blue light and then carry on again to blue sunlight. So effectively, again, the fish disappears. The eye of the fish determines how much the intensity of light to shine and controls that too. So that's a great camouflage mechanism. But then when we're getting mirrors also in the photophores themselves, the organs that produce the light, the bioluminescent light. This is one in a crustacean. These are the cells that make the light, and we've got a nice lens there to focus that. And behind that, we've got some black pigment to absorb anything that gets through the system, but also a nice mirror of multilayers here. And that focuses the light again. Any light that goes in that direction gets back reflected towards the lens. Here they are in the cephalopods, the cuttlefish and squid and octopus. And The red is the body of the animal, and the dots are the photophores producing the lights. I particularly like this one, actually. It's got one long tentacle that's got photophores positioned all the way along it. In the darkness, all you will see is a blue light going like this. And predators follow that and follow it all the way to the mouth of the squid and get eaten. Okay, now we've got the photonic crystals I mentioned at the start. These are things that now are turning up that technology really want to have, and they're trying to use more and more. They get used in telecommunication cables, but they're going to be used in optical chips too in the near future in computers. So when you've got the multilayers, we had periodicity in one direction, which is the direction in which the light is traveling, but then in other structures you can have periodicity in more than one direction. And that's when it gets complicated. Light comes in and starts to bounce around multiple times and interact all over the place in complex ways. And a good example of this is opal. So this is gemstone opal, and this is a cross-section showing what's causing those colours. It's a close-packed array of spheres about 0.5 micron in diameter. And then exactly the same thing has turned up in a beetle. This weevil here, these turquoise scales, those patches there are made up of scales, and if we chop one of those in half and look inside, we find that it's filled with opal. Exactly the same structure as the gemstone opal. And it's turning up in a whole range of these weevils. These ones are from the Philippines. And so we're trying to make use of this now. We're trying to copy some of the clever little properties that we're finding in animals in the ways that the spheres are held together in this case, but also in the amount of black that's in there as well. When we made this by packing together these spheres, it normally was very, very whitish, but we had a little bit of carbon black, and suddenly the wavelength's not reflected or absorbed, and the colour lights up. And then here's the inverse of that. These are weevils from New Guinea. This is the structure inside their scales, which is what we've called inverse opal, because we've got circular holes rather than spheres in the structure. But again, just the same principle. There it is in a butterfly. And this was one of those first animals I found that has the two-dimensional photonic crystal. This is a spine of Aphrodite or sea mouse. A polychaete worm. If you chop through that, it's a tube, and the wall of the tube is made up of 88 layers of tiny tubes, and that's a cross section through that wall. They're quite sophisticated. They have small diameter holes on the outside going through to larger diameter holes on the inside. Really, really difficult to model optically. There's the animal itself, another nice cross section. And these things are turning up all over the place. Jellied animals like ctenophores, cone jellies, as well as jellyfish. So yeah, our focus really now is on biomimetics and how we can actually make these structures. I mean, first of all, we have to make prototypes to show that the theory is correct, and actually that we can copy that color effect in commercial products. The one thing that's really been forgotten and doesn't actually show up very well here, it's explore the potential to optimally affect the eye. You know, we've been looking at technological applications, things that spectrometers read, anti-counterfeiting devices, to carry information such as in telecommunication cables or computer chips. But what about the fact that these colors have evolved to appear particularly bright to the eye, or particularly attractive to the eye? This is actually starting now to lead to a whole new series of applications that we're working on. There's the one idea is that, we've had a few conversations about this already, but the peacock's tail, for example, it was always thought to be brightly coloured so that it can, it relays some information of fitness of the individual. So a bigger, heavier tail, well, the individual must be stronger to be able to carry it. But we're now re-evaluating that and thinking, well, actually, maybe the female just prefers those colours. Maybe these things have evolved particular colours and patterns that elicit some kind of advanced response in the eye. So can we just take our colours and actually use them directly in applications, since they've evolved to look good? Here's one actually that's been used in a solar panel. This is a fly from Baltic amber, 45 million years old, and here's the surface of the eye, and it has fine striations. Now, we made those using a laser beam and fired lasers at it, and this is a smooth surface going from 0 degrees, that's the normal, through to 60 degrees either side of the normal, and we're getting significant reflection from the surface. So we're getting in the order of 5%, which is increasing with increasing angle. So that's the type of reflection you see when you look into a glass window and see a reflection of yourself. But this is the fly eye grating. And down to a 60 degrees either side of the normal, it cuts out those reflections almost completely. So we put this onto a window, we no longer see any reflection. But if you put it onto a solar panel, you get a 10% increase in energy capture through reducing any light loss through stray reflections. This is the moth eye structure, and this one here, and we've made that using focused ion beams. At the moment, we're using it to make a mould that can then be use more conventional methods to make it on a much cheaper scale. These are some of the beetle liquid crystals. There's the beetle, and that's its structure. And this is the structure we've made using titanium films. And these are some examples of the films. And they match the beetle exactly. As we move the film with angle next to the beetle, they change colour in exactly the same ways. This is the morpho butterfly structure that's been made using focused ion beam chemical vapor deposition technique. Here, this is what I've been doing in Shanghai. I've been replacing the original chitin material with titanium dioxide to make them more resilient, or zinc oxide. This is a diatom frustule, which surrounds the single cell and produces these colours. And this is what we've been making using deep photochemical etching. This is some new work we've just done to try to replicate a butterfly scale. These are the big ridges, and in between we've got the little optical structures. We are interested ultimately in trying to copy the manufacturing technique of the animals. It's one thing, okay, some of these have got novel optical devices that we can straightforward copy with our own machines. But if we can do it the same way the animals are, we are saving so much energy. They are making these structures at room temperature and pressure. We use huge machines to try to make the same thing, but using an amazing amount of energy, really high temperatures and pressures. Sometimes we're a bit stuck because we've been looking now at some of these using cell culture techniques to find out what's going on inside the cells. And the cell can be considered as a factory packed with little parts and engineering devices. So, for example, if we go back to this animal, this Sapphirina copepod, and consider that reflector, we then look inside the cell and we find the smooth endoplasmic reticulum looks like this. So it's very easy suddenly to make this device. You seed a crystal of guanine inside each of these compartments, and the crystal grows to the edges of the compartments, and that's it. So it acts as a nice mould to make these reflectors. Also considering what happens as a material dries, particularly in insects, and by nature they start to form different shapes. And that can explain some of these things here. So for example, you've got little fine holes here. These start out as thin transparent plates. But the interesting thing is that when they're wet and transparent, the whole butterfly isn't coloured. And then just a few seconds later, they shrink a bit and dry and crack in certain places to produce the holes. And you go from something that can't be seen to something that's visible from a mile's distance. And I think that's really interesting for biology, because you put this into a forest environment where everything's camouflaged, and it just suddenly creates chaos. Everything just suddenly has to adapt to this new light. Yet there was hardly any genetic mutation going on to go from a transparent butterfly to a blue one. So hardly any evolution, but the effect is absolutely dramatic. These are some results from our cell culture experiments, just a few. This is the morpho butterfly scale, the real one, from above and below. And here are the scales that we've grown after a different number of days. So after 0 days up to 22 days. And we can start to see at each stage the different structures of the final scale starting to appear. Right the way through until we get the final scale that looks exactly the same as it does on a living butterfly. Although it's not quite that easy. It took about 3 years to get those results. We also got these, we looked at the whole wing disc and showed that the pigment as well, the black pigment, because these butterflies, they need black to absorb those stray wavelengths, that appears after about 20 days. We've got, interesting is mimicry actually, because we've got this, this is a model of a mimicry ring. So these three here on the bottom are mimics of this species here. On the left is the normal white light, and on the right is an ultraviolet photograph. And this is the scale structure on the right. Now, one of the mimics has mimicked that exactly. This is really difficult because you've got a model in the environment, butterfly, that's warning birds that if it eats it, it contains a toxin. So the birds learn this colour equates with a toxin, so leaves that colour alone. But the signals are quite precise, and they can only be produced by specific types of scales, specific types of photonic structures. But how to evolve that really quickly before the model disappears, that's quite a problem for evolution. But it's managed to do so. And it's probably managed to do so because it uses those generic parts of the cell as molds and templates. So it can call upon these machinery at any time to make these optical structures. These are just a reflection spectra here of each of those to show that they do match really well. That's just to remind me actually, because going back to that idea about the peacock, is this really, these colours that are evolving in nature, the brightness of the colours, the signals that are within them, the polarization, the combination of colours that keep occurring again and again in all different species, are these things that the brain just likes? Is there just a preference for these things, and natural selection has taken advantage of that? So rather than the old peacock's tail fitness idea, maybe we just like these colour combinations. The reason this is here to remind me of these are Indonesian panels, ancient Indonesian panels that actually, there's lots of modern art today, that Rothko onwards, that actually mimic exact things. And the same, and you can take so many examples of this, go through different cultures that have independently produced these artworks, and they all look the same. We've got some cases of some American abstract artists mirroring some Aboriginal artists, producing exactly the same things in the same colour combinations and the same patterns. So really, is there something in this? It's notable also that those ones have become popular amongst all the artists that exist. That one I can't talk about, but just to finish off, just to as the latest news really, I mean, I've talked about the evolution of vision. Well, we assume that the first eyes saw colour, but we have no evidence. There's never been any evidence of colour vision in the fossil record. But now we've got a fish, we've just found one that's 300 million years old, and it's got very well preserved eyes here. And we found in the retina eumelanin, first of all, that we find in our own eyes. These top 6 here are the extinct fossil, And these are an extant goby that's related, a fish that's related to it, and you can see how similar the eumelanin is to that extant version. And here are the rods and cones in the eye. B and H there are a living fish, the living goby, that's the relative, but all these others are the fossil. And you can see that the rods and cones have preserved really, really well. So at least now we can say for sure that colour vision has existed for 300 million years, but probably it's been 521. And that's a couple of books you can read. Thanks very much. Yeah, now that's interesting too, of course, yeah. We just started looking at that because there was a species of bee from South America that's turned up that lives at very different a range of latitude, and from different parts of its environment, it's a different colour. We've also got a beetle from the south of France that inhabits different areas that are actually joined together in this large region of the south of France. But from each micro area, they appear a different colour. So there's evidence going on that there's something about the incident light conditions that might be affecting the colour that's being produced by the animal themselves. Of course, when it comes to pigments, they take colour quite often from the food source. So that's very much affected by environment. But in terms of structural colour, it shouldn't really be affected by the environment. Other than temperature, temperature can cause slight changes in the dimensions of physical structures and therefore affect the wavelengths reflected. But I think beyond that, I think, yeah, there's the properties of the incident sunlight are affecting the colours being produced.
And so then, I mean, we're affected adversely by blue light, but they are developing more blue, you say. Is that correct? Did I pick that up? That the color blue is now more, you see more blue in animals than you were previously?
I'm not so sure about that. We are actually doing some new tests at the moment using the museum collections over a period of 200 years because we've got really great collections of certain species of butterflies, for example, in large numbers, going back at least 200, sometimes 300 years. And what we're trying to do is equate that with changes in the light quality that's happened through climate change, for example, and seeing if there has been a direct effect of that. But we don't really have the answer to that. Now, in terms of blue, that was just, it turned out to be something that's really easy to evolve in an animal. But in terms of, say, the bees that I mentioned, they could easily evolve any colour they wanted because it's a quarter wave stack and you just change the dimensions slightly to produce the colour. But indeed, they have completely changed colour depending on the position within South America they exist. Yes?
I have a funny question, but as you were talking, thank you, as you were talking about the animals and how they adapt to color, I was thinking that human beings are animals too, and we have different skin colors and different hair color and eye color. Is this evolved? It started like one skin color and then it evolved to different skin colors, or?
Yes. I wouldn't want to comment on that because there are people who are particular experts in that. So I'm not sure what the latest argument is there, but I don't think so. I think that is more an adaptation to sunlight levels in different regions. So the amount of melanin that's produced in the skin. But, you know, there could be a case of female preferences have evolved, co-evolved with that, and possibly even taken over to be the overriding factor, and therefore driven evolution into that direction beyond what the environment would predict.
Concerning evolution, Andrew, I remember in your book you showed an example of how Darwin had this big problem about how such complex structure as an eye had evolved, because there's no direct step from the beginning to the complete thing that has to be all together to function. And you show some example where, with I don't know, maybe 10,000 steps, very small steps, within 1 million years you could conceivably evolve the whole structure.
That's right. Yes, yeah, no, absolutely. So, yeah, I mean, Darwin himself picked out the eye as a problem to his theory of natural selection because he thought that, you know, he saw the eye as something extremely complex and perfect, as he called it. And he thought that, how can this evolve? Because half an eye doesn't work. So how can you get stepwise to that fully evolved eye? But it was soon shown that not only can you get to the eye, a fully evolved eye from just a sensitive patch of skin, a light-sensitive patch of skin, to a fully developed lens eye such as our own, in 300,000 generations, which is, 300,000 years in a lot of animals that we're talking about. Not only there's actually not that many steps, but in fact each step does provide an advantage over its predecessor. So you go from just detecting the direction of light to becoming a sort of a more convexly curved surface to more accurately detecting the direction of light. And that continues as you increase the curvature, you increase the sensitivity of the directional sensor. But there's something that still needs to be solved because you've got this almost pinhole camera type eye structure now. You've got this inverted sphere with a little hole in the top for light to come through, which detects very well the direction of light. But the next step, which Richard Dawkins actually just threw in as another step, is the formation of a lens. Now that actually increases the amount of information you receive by 1,000 times. So it looks like one step on that nice little chart, but actually it's not. It's something that suddenly blows the whole thing out of the water. What goes on there? You suddenly, how can we cope, or how could that animal cope with suddenly having 10,000 times more information? And that's one thing we've been working, we produced one paper that the answer there might be sensory substitution. So it started to borrow cells from another sense, such as the sense of touch. You know, in the way that if you take a blind person and shine a pattern of light onto their skin or their tongue, they perceive it using the part of vision otherwise used, part of the brain otherwise used for vision in a sighted person. So the cells are very plastic, the nerve cells. You can move around from sense to sense. So maybe they borrowed it from another sense. Or maybe it was just multiple substitutions. Maybe they just took the nerve cells and part of the brain that was adapted to vision and multiplied it through genetic multiplications many, many times.
Because this neat progression of 300,000 generations, it works from beginning to end. If everything goes exactly in the right direction every single step. But what do you make of arguments like Sheldrake quotes certain scientists who show that statistically it's nearly impossible that such a thing would actually happen? For you, it's not an issue if you consider that it's.
Well, I think that I think the numbers. If it was just one individual, then it's going to be very unlikely. But if you think that this experiment is repeating millions and millions of times, then eventually the populations that survive best are going to be those with the enhanced visual receptors. Yes.
There is such a thing as tetrachromacy.
Yes.
And it is known that certain very gifted females have tetrachromacy. So evolution is still going on. We're not perfect yet. So could you comment on that, please?
Yeah, I mean, absolutely, yes. I mean, where's this all going to end? I mean, there are some species with 19 visual pigments, some stomatopods, for example. But that's right, yeah, maybe we are going to become increasingly colourful, and if it actually results in an increased number of offspring, then those genes are going to be retained, and there are going to be more and more of them in our populations, and eventually it'll become dominant. Sorry,
eventually overpopulation.
Well, absolutely, yes, yeah, that's right. Then we'll need to be wearing lots of bright colours.
Are there any new insights from epigenetics concerning sight and colours?
Yeah, that's something really I haven't been involved with. So yeah, I can't really answer that. But the eye has been a target in those studies. So I think that yeah, it's worth having a look at the new literature I think that's coming out there.
Any other questions? All right. Thanks very much.
That's great, thank you.